molecular biology.
Recording in progress.
The first one is the transcription. First process is the transcription. The transcription is how DNA is transcribed into mRNA. So we have data, the genetical data stored in DNA as far as the humans are concerned. Right? Deoxynucleic acid, that is what the DNA. So the DNA to the middle man or the messenger, RNA. This process, the conversion of information inside the DNA to RNA, is called the transcription. Then translation is how this messenger RNA translate this information at the protein synthesis factory to proteins. So there are two steps in which the information get translated. This is like, this is one person understand one language, is another person understand this language A and another language B, and this person understands only language B, something like that. Language C. So this person can't directly talk to this person. I'm extremely simplifying this, right? So there is a interconversion, translation in the middle. So you first convert the information into mRNA, the messenger, then the messenger converts that information into proteins. And this is called the central dogma of molecular biology. And we spoke about the biological data types. We'll go into this in little details today. Now we want to look at the structure of the DNA. Right? I'm not planning to do this as for a biology lesson, or as for a biochemistry lesson, or as for a chemistry lesson. So I want to present this information so that you guys who are lacking the background in biology and biochemistry can understand this, right? So it's a, if you think about the DNA molecule, it's look like this. It's a what we call a double helix spiral. Have you seen these staircases sometimes that are outside the buildings, right, to give access to the upper floors, but from outside it goes like this spiral, right? So exactly like that, you have two bars like this, and then you have steps in between, right? Now you know how does the DNA look like, and then each step on that staircase, if you take a step, if you take just a vertical part of your staircase, it's got things like steps like this, right? And then it goes like this. But each step now here has two components, one on this side, another one on that side. So if you look at the diagram here, let me zoom into this. Now you can see the structure of this double helix DNA molecule. Now we have different types of molecules forming the crossbars or the steps here, and they are named and they are labeled using a single English letter, GCTA, standing for guanine, cytosine, thymine and adenosine. These are molecules, right? And their structures are given here on the other side, right? So you can see this is how the adenosine looks like. It has a certain chemical structure and a glucose part, and here's the thymine, here's the cytosine, and here's the guanine, right? So what you have to understand as computer scientists at this point is Okay, so there are four different types of molecules that make the DNA structure. They are adenosine, thymine, cytosine, and guanine, and then they are given one letter code A, T, C, and G. Then, if you think about, let's say that these two, the staircase, this, let me go back to the structure over here. The spherical staircase come in two parts. Now you want to assemble them. So it comes in two halves. So if you divided it like this, in the middle, it comes in two halves. Right? Now you want to put them together. Right? It comes together. Now you want to, when you put them together, you want to make sure that there is a specific, unique pairing between these building blocks. So A has to always combine with T, and T has to combine with A. C has to combine with G, of course then G has to combine with C. So you can't combine A and C together. It won't fit. And chemically it won't fit. Then it will not give you this correct spiral shape of the DNA. Now why this is important to us? Because we are going to look at this sequence. Right? This is called the sequence. DNA sequence. If you take this part of the DNA molecule, the sequence A, T, C, G. Right? A, C, T, G. Now we have a long DNA strands like this. Where is this? This is inside the cell. Right? Inside the cell, this is the human cell. Inside the cell we have a specific part called nucleus. That is the controlling panel of the cell, if you want. Right? It's a different compartment, what we call the cellular compartment. So nucleus is a specific cellular compartment in which we have the genetic material, in this case DNA, and this DNA is found in chromosomes. Right? The chromosome is nothing but a DNA molecule wrapped around with a protein. Right? It's a chromosome. If you think about a sausage now, right? You have a sausage wrapped around with a thin cover. Right? It looks like your chromosome. You can think about any other example. Now inside that you have DNA. So this picture shows you where the DNA is inside the cell. So the DNA wrap around in a protein coating, and then this protein coating, this coils like this to give you specific chromosomes. Right? And why they are given the name chromosome? Because initially when they looked at these cells under microscope with certain chemical color, different components of the cell, this get an intense color. Chrome means color. Right? So they could see this specific units getting colored with their chemicals under light microscope. So they call them chromosome. Now what is the relationship between the chromosome, DNA, and the most important part to us is a gene. Right? So now we know the relationship between the DNA and the chromosome. DNA is like a double helix. We call the shape is a double helix. Double helix shape. Like this staircase, as I mentioned you, so it's a double helix. And then it's composed of 4 different types of molecules: adenine, cytosine, guanine, thymine. And then these cross bridges are formed between 2 parts of the bridge. A and T and C and G must be paired. Right. Then These DNAs are wrapped around with a protein coating, and then we call the chromosome, and then the chromosomes, many chromosomes are found inside the nucleus. Right? So when a male cell and the female cell fuse together, right, they have different chromosomes coming from um, males and female. Right? For example, there is a gender determining chromosome, and that is called XX in female and XY in males. So if XX is a female and XY is a male, if X and X from a female and X from a male combines, then you have a female. Sorry. Then X from the female and Y from the male combines, then you have XY, then you have a male, right? So this is how the gender determination happens based on the chromosomes. Now, why are we studying this? Because in the DNA sequence, 3 units of these sequences is called a gene, right? So if you search for the definition of the gene, now let's say, so let's look at the what is a gene. Just Google it and see what's the definition of a gene. So it gives you the gene is the basic unit of heredity. The molecular gene is sequence of nucleotide in DNA that is transcribed. Where is it now? So how many units are involved in a gene? 3, right? So we have different genes responsible for different things in our body. So it is important for us to understand the entire DNA sequence, and then in this DNA sequence we have genes, and then each gene is responsible for coding one polypeptide chain. Polypeptide chain is the if you want, you can very loosely understand each gene is responsible for one protein, coding one protein. So it is important. This is where the relationship is coming from between the DNA sequence and the proteins that we have in our body, right? So sequence is converted and given into mRNA, and we make the protein. Now in bioinformatics, what we are trying to do is, first we are trying to identify the DNA sequence, then we are trying to identify the genes, then we want to see if there are problems in a given sequence, then we want to do sequence matching, right? Then we want to do sequence alignment for various reasons, right? So to do that, we need bioinformatics tools, computational tools. Now for example, at the end of the day, what you have is a combination of 4 letters: A, C, G, T, right? Then I keep going on. It's another T, another C, another G, right? Another T, right? C. Now what is this? To you as a computer scientist, this is a sequence of letters, right? Then if I give you another sequence of letters, you should be able to do what? You should be able to do sequence matching. Do I have any matching regions between these 2 sequences? So I give you one sequence like this, then I give you the sequence 2. Now can you write a piece of code to see how similar these 2 sequences are? Do you understand what I mean? Right? So you give one sequence as your input, another sequence as your input, then you want to compare S1 and S2. Then not only you can, when you do the comparison, you can start like this, starting end-to-end comparison. What else can you do? You can also keep an offset. You can start from the first one and first one compare, and then the rest of the sequences. Then what you can do, you can keep an offset. The first one of the first sequence is now is aligning with the third one of the second sequence. So there's an offset. Then there will be more matching. You don't know, right? So likewise, the sequence comparison, pattern matching, becomes very important in bioinformatics. So why do we do this? We want to see, for example, if these two DNA codes are, how similar they are. So maybe we can finally use that information to relate these two living creatures in terms of their evolution, right? Then maybe it is the sequence from two different virus variants. Now, when there was COVID-19, because the virus was evolving, its genetic material was evolving constantly, right? So we had the gamma, mu, beta, whatever different variants. Why? Because they are sequence. In virus we have RNA, not the DNA. We'll see what RNA is, right? But it's the same sort of scenario. Then if the genetic code is changed, the protein that they produce is also changed. Then therefore their behavior is also changed, right? They can become more virulent, more problematic. They have more strength, right? They can hide from our immunity response. They can hide from our immunocells, right? So that's what they did. So they constantly throughout this corona period, they did the sequencing of the virus, different variants. So then you have seen in the news, they say that they have done the sequencing for the new virus, and it is 80% similar to the previous one, but there are certain different changes in their primary sequence. So how do they do that? They get the sequence and then do the sequence comparison using the softwares, right? So this is the significance of this information. Okay, right. So as we spoke here, the significance of the DNA structure: adenine, thymine, cytosine, and guanine. These are the molecules. If you want, you can forget about the chemical structure. I'm not interested in teaching you the chemical structure of it, right? What we are looking at is from more computer science point of view, and how this information is useful to us when we write codes and programs, and then how can that data be interpreted and analyzed to retrieve important biological information. So you have 4 building blocks, and then they pair with their relevant partners, right? So DNA sequence of bases A, T, C, and G encodes the instructions for building proteins, which perform the most of work in the cells. That's the essence of life, in other words, right? So the double-stranded structure provides a stability for genetic material. So these strands are separated during DNA replication, allowing for accurate copying and genetic information. So if cell divides, you need to copy the genetic material, right? Because you know that, for example, your skin cells, right? The lifetime of a skin, the top layer of the skin cell is about 30 days, right? You can see your nail grows, right? So these cells do have a limited amount of lifetime, but underneath there is a new layer coming up, right? So the cells need to divide. So cells need to divide, therefore the genetic material need to be copied accurately. So in the copying process, if something goes wrong, what will happen? Then you have a problem. Sometimes the signals are messed up. There is a signals. There are signals to stop reproducing cells, right? The cell death must be there. So you, excuse me. So you have a cell. This cell keep dividing, and then okay, you have a tissue then. So at one point this cell division must be stopped, so you have the correct number of cells. But under certain circumstances these signals are messed up, and then these cells keep growing forever, and that is a what? That is a cancer, right? Because cell dividing signals are messed up. It keep dividing forever without a control. Okay, and then there are certain genes get expressed. Certain proteins are made in excess in cancer cells, for example. Then if we are in a position to identify that, we can find out probably what is the reason for this particular cancer. Right? So that's what people are doing. So they're taking samples from RNA samples from these cancer cells, and then do what we call the gene expression analysis to see what genes get overexpressed and suppressed in cancer cells compared to a healthy cell. Overexpressed in the sense gene gets overexpressed. So gene is a code in your DNA. Now you have a DNA sequence. You have a 3 block segments, which is a 3 blocks, 3 base pairs, 3 blocks is a gene. Each gene is responsible for different proteins. So this gene, for example, codes for the protein 1. The information that is required to make protein 1 is in that particular gene. So that gene is given the same name as P1. So P1 gene. So P2 gene, for example, make the protein P2 likewise. Now in cancer cells, for example, in this particular cancer, you will see there's lots of P1 proteins, huge amount of P1 proteins. That means this P1 gene is overexpressed compared to a healthy cell. Let's say, for example, in a healthy cell, you are supposed to have 100 units of P1 protein P1, but in a cancer cell there's a 1 million P1 cells, right? So then we want to find out why and how it happens, then what is the relationship between that overexpression and this cancer and all that, right? So we need to analyze this data, gene expression data and genetic data. Now the next section that we are planning to get into is the biological data and databases, right? Okay, so as we said, where do this biological data come from? It comes from genes, right? DNA sequences, genes and genomes, and it comes from proteomics. Usually the data that we handle in bioinformatics is called omics data. Omics data. The omics, because we have omic data, right? The data comes from genetic material, DNA, genes, and the genome. The genome is basically, we'll see what it means. And then proteomic data, once again omic data, the protein sequences, structures and functional information. And we have transcriptomic data, and that is basically the data that is coming from the RNA sequences and gene expression data. And other data types, we have metabolomic data, right? So omic data, in general we call them omic data. So how do we gather this omic data is from experiments, right? So we looked at the gene expressions, we looked at the protein sequences, we look at the protein structures, we look at the metabolic pathways. The experimental scientists in their laboratories, they collect omic data, right? And what do is we collect this omic data and then try to analyze them using special software and bioinformatic tools to identify patterns and then interpret them in a biologically meaningful way. Um, I think you've learned this in that small video that we watched last time, right? Omics data, right? And the characteristics of biological data. Biological data is complex and it's diverse, right? There's a huge amount of data, it's complex data, and it's different types of data from one to another. So we will be looking at the complexity and the diversity of the biological data as well. Okay. Okay. Sorry. Do you still? I didn't see the message. Do you still get the background noise? Sanura, is my sound not very clear? Okay. Just hold on a minute. Let me see if I can address this problem. All right. Now let's see. Is it better now? Is my voice clear and better now? Can someone please respond? Bit clear. Ah, okay, right. So I'll try to speak a little bit louder. Okay. Okay, sorry about it. And where are we? Yeah, the omics data. And then uh, we want to talk about the data formats in bioinformatics, because when we store data, there should be a consistent way of storing biological data, right? For example, genetic data is stored in a particular way, and the protein structure data is stored in a particular way. We'll have a look at maybe a protein structure file, and then see what the data format is. So because now you want to write applications that uses this information to interpret them and to do other calculations that are required in bioinformatics. And then also data quality and annotation. So we want to have quality data and then annotate it, maybe highlight it, and then basically tag them with their specific role in structure or function or biological pathways. So highlighting is called the annotation. So we'll see the genomic data, right? So the genome refers, sorry. So the genomic data refers to the complete set of DNA, including all of its genes within an organism. So human genome, we spoke about the human genome project in the last lecture, where we looked at, not we, I mean, of course, the scientists who involved in the genomic project, human genome project, they looked at the entire set of genes and then they mapped it. So that means now we know what gene is responsible for making what protein. So now here I try to sort of give this idea in a picture like this, what I found on the internet. So you can see there's a huge amount of data coming from this piece of DNA only, because each as I told you, 3 on your DNA segment, 3 blocks, 3 building blocks is a codon. This is given a special name called a codon, and that is responsible for making or coding one amino acid. So if you look at the protein now, how does a protein look like? You are eating proteins on the regular basis, right? You are eating meat, you are eating fish, right? It's protein. Okay, so the proteins, different proteins do have, as we spoke last time, they have different roles in our body. So proteins are made out of, their building blocks are called amino acids, right? So initially, when the protein factory inside our body makes the protein, it looks like this. Every protein. So this is like a necklace, that is where you have beads on a thread. Each bead is called an amino acid. It's once again, these are molecules, right? Amino acids. Amino acids. And in nature there are about 20 amino acids, not that many, but there are about millions and million different proteins, all composed of these 20 amino acids. So how come? Because their sequence is different, right? So if you label these amino acids, just forget about their chemical names, right? To you what matters is just the labeling, right? So they have different names. So different amino acids may be they have different let me get to something different here. Okay, hold on. Single letter amino acid codes. to get something onto the slide. Hold on. Back to my slide over here. Right. Okay, now can you see my screen? Yes. Okay, so now I have shared you the names of the amino acids that are there out in the nature: alanine, arginine, asparagine, aspartic acid, cysteine, likewise. And then they have usually in chemistry and biochemistry, we use the three-letter symbol, right? That is in the second column. But when it comes to bioinformatics, it's very easy for us to have a single letter code, because when it is a sequence, each letter refers to one particular amino acid. A is for alanine, R for arginine, likewise. Now I was saying that when the protein is made, so you have a protein synthesis factory, what we call the ribosome in the cell. So the mRNA, the messenger brings the information from DNA. Then it is given to the ribosome. Then the ribosome assembles these amino acids according to the information in the mRNA, the messenger RNA, which was originally in the DNA. Right? So this is how this information is converted. Now each amino acid, and then have the first amino acid, then the second one is added, then the third one is added, so on and so forth, right? So as soon as the production is over, your protein looks like this. So this is called the polypeptide chain. Polypeptide chain. Now this has different amino acids sequence. So this could be A, D, E, C, A, L, I, P. What are those? These are different amino acids. Now they are connected together, right? Then in the body, what will happen is this chain will fold it into a three-dimensional shape, right? Let's say, have you seen the baskets that are made out of metal wires, right? Baskets and then flower vases made out of metal wires. You have seen them, right? So you take a single metal wire, so there's no particular shape. It is not functional. Now you can fold it, wrap it around, fold it in a very specific way, and then you can make a basket out of it, right? So then that becomes functional. So this is the three-dimensional functional form of the protein. Then folding this primary wire chain type of thing into the functional form of the protein is called protein folding. Right. Now it is for us to understand the sequence. Right? We are mainly worried about sequence and sequence analysis and pattern matching and all that in bioinformatics. Now I want to see, I know the primary sequence. I have isolated a brand new protein from a new virus, right, which is not known to mankind before, something like COVID-19. So this is the sequence in one of the proteins. Now I want to see what are the similar proteins to this particular protein that we already know. Hold on. Some of you can't see the screen. Is that true? Hmm. Hold on. Now how about this? Sorry about it. Okay, so let me. Okay, sorry. Let me go through it again. I'm sorry about it. Um, I was saying that the the protein making factory is the ribosome, and then ribosome gets the signal or the sequence information from the mRNA, who got it from the DNA. So this is the DNA originally stores the information, then that is given to mRNA, then mRNA brings that to the protein factory, which is ribosome. Then the mRNA goes through the protein factory, then this reads the sequence on the mRNA. So according to the information on the mRNA, it brings in the correct amino acids. So the first one is A, and then which is connected to next one is D, and it connects it. It's like you are making now a necklace with different beads, right? So that is what the ribosome is doing. And then this next one is E, C likewise. So we have this, these are the one letter codes for these amino acids, right? So it's a particular, we call this the polypeptide chain. Then the polypeptide chain folds into a three-dimensional functional form. As I just said, this is just like a single wire. You can fold it into different functional forms, right? So here we have a three-dimensional functional form. Every single protein in your body has a three-dimensional functional form. Now this three-dimensional shape that it gets into finally is mainly determined by the sequence over here, right? So if you know already certain sequences and their corresponding three-dimensional shapes, you can predict a new sequence and its three-dimensional shape. What I mean is this. So let's look at this scenario over here. Now let's say you have some people. They are in a line, but they are connected to a rope like this, right? Some people, they are connected to a rope, and they are all connected. Now over the time, what's going to happen? They're going to make friends, right? These guys going to talk to each other. This guy will talk to this one, that guy will talk to that one, this one will talk to this one, right? So they make interactions. The same with the molecules. And then after a while, how it's going to look like? It's going to look like this. So they all are making interactions with one another. Now they have got into a different form, right? So the shape, this, if this is happening in a 3 dimensions, so this will be a 3-dimensional form, a structure, shape. Now it all depends on who talks to who. Maybe this guy likes this one, but he doesn't like that one, so they don't want to talk. So the third one tries to stay away from the fourth one. You understand what I mean? So then the final shape that you get depends mainly on who is sitting next to you. Okay, so there are experimental ways of getting the 3-dimensional structure of the protein. So let me first show you what I mean by a 3-dimensional structure. I think I have a picture here. Now can you see this picture, the blue color picture? That is a 3-dimensional structure of a protein, right? So it has a very specific shape. Now if I want to outline the shape, sorry, I will show you basically. Let me now do it as we speak. So let me first show you a structure, then you will understand what I mean. Okay, give me 2 minutes. Let me get my application ready. um, okay. Bad luck's got stuck here. Yeah. Okay, so this is the protein structure. I'm going to change how it looks like. We going to use the too many things happening here. First, we look at its three-dimensional structure. Can you see this structure? Yes. Okay, good. Now I can rotate it. Right, this is a specific application to visualize biological molecules. Now you can see it's a three-dimensional shape over there. Right. So this is the functional form of a protein. This is how a protein would look like. And then we want to represent, if you want to see the amino acids in that, I can these are different types of amino acids in different colors, right? You can see there are brown. These are the ones that we discussed: A, I, P, L, lysine, arginine, type of things. These are amino acids, different amino acids, right? So if you want to look at how they are connected, we can still change the see, is the actual chemical structure. So they have formed into a functional, folded into a functional form. See, these are molecules, right? Amino acids. Each color different color is a different amino acids, right? So you can see they are connected to one another, right? So that I schematically represented it with a sphere, with a bead, right, on a string. So this is the string now folded into a different 3D shape, right? So what matters to get this three-dimensional shape is the sequence. Now we have an assumption in molecular biology: similar sequences will have similar structures, right? So now let's say getting the three-dimensional structure is a very important task in biology, in medicine, right? Because if you know the three-dimensional structure of your protein, then it is easy for you to understand its behavior, its other functions, and how to stop, if you want, how to stop its functions. That means, in other words, if you want to design a drug molecule to stop the action of um, this particular molecule, what a pharmaceutical drug usually is, a molecule that blocks a protein like this, right? So if you take paracetamol, Panadol, when you have a pain, when you have a headache, so there's a pain signaling pathway inside your body, and there are certain proteins involved in this pain signaling pathway. So paracetamol goes and blocks one of these proteins, so it can't participate, it can't do its action in the pain signaling mechanism, so you don't feel the pain. You take aspirin goes and stops another protein. If you take a powerful other painkiller like celecoxib or Nexium, or not Nexium, there's a Neocoxia or something like that, very powerful painkillers. So they go and attack different proteins, right? So knowing what I'm saying is, knowing this three-dimensional structure is very important to us. So the point is the primary sequence. Now let's say you know this structure and you know its primary sequence, but now what we can do is use that assumption of similar proteins will have similar structures. We can use that assumption to determine the three-dimensional structure of unknown proteins. So these structures are obtained by using, for example, this particular one is obtained by using experimental technique called X-ray crystallography. Like, you isolate the protein, you make a crystal, a solid crystal of that protein, like your salt crystal, and then send an X-ray beam through it, and then you get the structure. The same as you getting a X-ray picture of your broken hand, right? X-ray picture. So X-ray crystallography is one technique of determining the protein 3D structures, and there are other techniques such as NMR techniques, something similar to what they use in MRI scanning, right? The same technique they can use, you can use that to determine the three-dimensional shape of the protein structures. But in certain cases we can't do that. Then my point is, now we go back to our slides. Now my point is this, what is happening to this? So it's not erasing the content of that. Hope you can see my screen now. Yes. So you have one sequence, this particular amino acid sequence, and for that sequence you already know the 3D structure, something like that we looked at before. Then you have a brand new sequence. It's called the query sequence, SQ, new sequence, and you don't have a 3D structure. Now what you can do is you can use bioinformatics tools. You can write a piece of code to look at the structure, the sequence similarity, right? So you can compare these sequences and then identify the sequence similarity. Now using your programming skills, can you do that for me? Just forget about the amino acids. You get this string, right? So I'll give you like a piece of sequence. Sequence 1 would be A R N D C E C C I L K. 1 2 3 4 5 6 7 8 9 10 11 12. That's the first sequence. Then the next one is N N R N D C E C I A R. 1 2 3 4 5 6 7 8 9 10 11 12. Now I have two sequences. Now as a programmer, can you write a small piece of code to give me the sequence similarity? What is the percentage similarity between the sequence 1 and sequence 2? We call this sequence 1 and sequence 2. Can you do that? How long it's going to take for you to do it? So you are programmers, right? Efficient, capable, competent, confident programmers in Java. This is what we expect from you guys as programmers, software engineers in bioinformatics. You should be able to give us the codes, the tools. Now you see directly where your software engineering skills go into bioinformatics. This is a real life requirement. How many of you can do it, you think? Hands up if you think you can do it. Yes, you compare the sequence, okay. And then I need a percentage similarity. And then I want you to identify the regions. Not only the first requirement is the percentage similarity, sequence similarity. Not only percentage similarity, I want you to show me the matching segments. Right? So you have two outputs. How long it's going to take? 5 minutes? Ravindra Pushpakumar, how many minutes do you want? And also Indika Udaya Vikramarachi, yes. How many minutes do you guys want? 5 minutes, 10 minutes to get the code? Now you know the requirement, right? So what is the importance of knowing this? Let's say you can do it as a homework, right? Write a piece of code in Java to compare these two sequences and give me the percentage similarity and then the matching segments. So especially knowing the matching segments are very important. Why? Because we have the assumption. What is the assumption? Similar sequences will have similar structures. So now, for example, you look at this segment over here. RNDCEC, this segment is matching exactly to that segment over there, right? So what would be the conclusion? The three-dimensional shape of this region, let's say this region looks like this, then this region is also going to look like that. So if you have this kind of tool, you can use that tool to predict the three-dimensional structure of a brand new sequence. And this is in fact the essential concept in what we call homology modeling. Let me write it properly here. Homology modeling. Homology modeling of protein 3D structures, in which we use a known three-dimensional structure and its sequence, and that is called the template. Now what you have in hand is a brand new sequence whose 3D structures are not known. Sequence 2. This is your query, the brand new sequence. Then first objective is for you to identify the correct template, right? So in order to identify the correct template, you have to go through a database of structures, proteins, whose 3D structures are already known, and of course their primary sequences are known. Then you need to compare and match your sequence to every single sequence in the database and then get the best one out of it. So that is why we wanted the similarity, percentage similarity. If you want to use one protein as a template, we say that we need to have at least 40% of sequence similarity, at least. If your template and your query sequence are having less than 40% of similarity sequence, you can't do homology modeling. Okay. Now, then what is the next challenge? So I have a database of protein structures. This is the database of protein structures, protein database, where I have now, as for 2024, as for today, there are about I think nearly 300,000 protein structures, right? So 1, 2, 3 up to 300,000, let's say, is a huge database. And then on the other side we have another database called Uniport. Uniport is the world's largest sequence database. So you can use Uniport to first identify the proteins, right? The sequences. Now what you need to do? You need to give your sequence to Uniport or to protein data bank, and then first identify the template. So if you want to use Uniport, then it's a two-step process. You have to use Uniport and then get the template and then check in the PDB whether you have a structure for that template, or you can directly submit your query sequence to protein data bank, and then what would be your output? Your output is a template who's got at least 40% of similarity, sequence similarity. Now how are you going to do this? Now you are the computer scientist, you are the software engineer. Now you see directly where your computer software skills and knowledge goes into real life problems in bioinformatics. Right? So that is why I told you right from the beginning, we are not trying to uh, learn this course from the biological molecular biology perspective, right? We are more worried about the computer science software engineering perspective of bioinformatics, right? Because this course is a bioinformatics course for computer scientists, so you should know where to apply your knowledge directly in this particular field. Now I've given you here two examples, direct examples of where your knowledge goes into. Now I'll show you the protein data bank also, PDB. Let me stop sharing here. If you now I think you can see my browser, right? Yes. Where's the rest of the students? I can see only 3-4 people. Have you gone for sleep already, or having your supper, having your dinner? Already sleeping? How many lectures did you have today? How many lectures? Is it the first lecture for the day or what? Hurry up, talk. This is the only one. So why do you look so tired and so worried? Huh? So where's the rest of the class? Can you switch on the cameras, please? Are you playing games, or are you talking to your grandmother, or what are you doing? Huh? Is your grandmother feeding you while you are listening to the bioinformatics content? Okay. So okay, someone did it. Kavinder, right? Kavinder Satsar. Percentage similarities 45.55. Matching segments. Okay. So what is the code? Can I see the code, please? Okay. So thank you for doing it. Right. So then calling the Uniport API, and then searching for our sequence, and then call PDB API, and then get the results. Okay. Right. Good. So you have some idea about how to do this. It's very important. Now let's say this is the protein data bank. So world's largest repository, the deposit of protein structures. Right. There are about 24,000 roughly the structures from the PDB, and there are about 1 million computed structures. That means computationally generated structures using bioinformatics tools. We will see how we could do this maybe in the next lecture, right? How we could generate a three-dimensional structure from primary sequence of the protein. Getting the sequence is not a problem, right? Usually the problem is getting the three-dimensional structure. Now, even during the corona period, they found the virus, then they isolated the proteins in the virus, then they got the sequencing, because you can express different proteins in yeast and other cells. You can cultivate the protein in other creatures. you can use some other creature's protein synthesis mechanism to produce someone else's protein. So that is not a problem. Then you can get the sequence. So the sequencing in general is not a problem, but the problem is to get the 3D structure. So here you can search each entry. Now you need to understand the structure of the database first before you write a piece of code to access the data inside the PDB. The structure of the database and the security issues, right? So that's important with any database. So each entry in protein database has unique PDB ID, right? So if I type like COVID-19, you can search with keywords, and all the proteins related to COVID-19 are given here, right? SARS-CoV-2, this is COVID-2, right? RNA, right? So here's the SARS COVID 2.5, COVID-2, 5' proximal stem loop. There are different RNA segments over here, right? And let's say let's take a different more what we call this. For example, let's take this one. So the ricin is a plant protein. It's a toxic, plant toxic. Here's the structure of it: crystallographic refinement of ricin to 2.5 angstroms. That simply means the crystal structure, X-ray structure of this particular protein called ricin. Now, there's always a 4-letter unique PDB ID in this database. Then you can go into that particular entry to retrieve more information about this particular protein. Now it's got the structure over here, then here we got the sequence annotations. Now you see, and then it has the uniport sequence also. This has amino acid residues. For example, binding chain is over here. Now as we move on that, you can see the binding chains get highlighted, and binding chain, buried residues. Buried residues means residues that are deep inside the protein. Now you can see when it comes to this side, you see these are inner type of protein. This is the, if you see, sequence annotation. So we have highlighted. This annotation means you have highlighted different features of different parts of the sequence. Now you can do that in your database. Then you also get the normal, let's say we go back, and then the actual sequence over here. This is not what I want. Where is the actual sequence? Sequence, not sequence alignment. Hold on, let me see. We have the structure summary, and then the publication in which this was published originally. here's the sequence. So you can see as I move the cursor over the sequence, it is showing you the relevant part in the protein structure. So each letter, these are one letter notation for amino acids, right? So now this is given, this has a 3D structure composed of different amino acids. It's got 260 odd amino acids. So you can see as I move on where the specific place here. Now if you want to use this as a template, of course you need to use this sequence as your template and then compare it with your new sequence. Then when you do the sequence matching, once again in your algorithm, right, when you do the sequence matching, let's go back to you want to be careful that you are doing it in different ways, not in hold on. Okay, as I told you, you can do the sequence matching starting with the sequence 1, sequence 2. You can align them like this. So this is called the sequence alignment, right? So before we start doing sequence matching, you have to do a sequence alignment. The alignment can be like one-to-one alignment, starting like this, or you can have an alignment with an offset. So the first amino acid of this is not just aligning with the first amino acid of the second one. So you can have like this. So this is the offset. There's no matching region for that, right? So you can have different offsets and see if the matching improves, right? So in first case like this, you might get a 25% matching. But if you, when you have an offset like this, you might get 55% matching. Now how do you do this? How do you know which one is to which way you're going to do the sequence alignment? So then you have to have a sequence alignment algorithms, right? A particular systematic way of doing the alignment. So that's here's the third application for you to try, right? So take a lengthy piece of polypeptide chain, a sequence, and then have different offsets and then see how the matching improves, or maybe it could get worse, right? So then you have to do this with a single entry. You have to do multiple alignments, right? The multiple sequence alignment, that is called the multiple sequence alignment. You can align multiple times with a single structure, or you can align your same structure with the multiple different sequences, right? So therefore, in order to do either, you have to have proper sequence alignment algorithm. So the next challenge is to come up with sequence alignment algorithms, right? So as software engineers in bioinformatics. Now we look at different types. We're still talking about the genomic data, how big that could be, and what's the importance of it, and how we could use that information to come up with new structures. Then how big is this data? So it is predict genomic data, or the genomic projects will generate 40 exabytes of data in the next decade, right? So 40 exabyte is about this much. You have different sharks. I got this picture from the internet, and each shark is about 100 million gigabyte. and you can calculate how many sharks here. So this much of data will be produced in the next, this is a prediction, right, in the next 10 years or so, in the in the genomic projects. This is only genomic data only, right? So this is huge. So that is why you need a proper way of storing them, accessing them, efficient algorithm. If you want to go through this amount of data, your algorithm needs to be very efficient, right? So these are the main challenges. You can think about this is becoming now big data. So we can talk about a separate topic on big data in bioinformatics, right? Maybe I think there are books even written on that topic. So this is becoming handling of big data, which is basically a separate, I mean, it's a different art. I'm sure that you have had some lectures on big data, right? Yes? No? You didn't have any sections on big data, big data handling and data mining and all that, right? So it's a different art. Okay. So human genomic project, and then what is the significance of it? We can use this information in genetic research, personalized medicine. It's very important because if you analyze your genomic data, one advantage that we have is we can predict whether we're going to have any genetic diseases, or whether we are about to have any other type of diseases as we move on, right? So now specifically this technology is to a certain extent available in Sri Lanka, where recently I saw an advertisement by the Asari hospitals. They do this for couples. If you are about to get married, you can get your genes analyzed and then predict whether you're going to have children with genetic problems. There are diseases, there are genetic diseases, mainly Down syndromes and things like that, right? Or whether you to have genetically problematic offspring, children. I think it's better to do it that way other than looking at your horoscopes, right? Because it's more scientific. So if you're planning to get married, it's a good test to check if your baby is going to have any genetical problems, right? And then not only that, even if someone is having a cancer, you can look at and analyze their genetic information and then look at specific treatments, because even the same cancer manifests in different uh, ways in different patients, right? Because they have slightly different biochemical composition. So your biochemical even though we are all humans, we belongs to the same species, your biochemical composition is slightly different to mine. Okay, so depending on what genes are expressed it to what extent. So if someone can analyze that, then you can have a very personalized um, medicine or medicinal scheme for certain diseases. Or we can also use this genetic information to look at the evolutionary relationships. For example, if you analyze the genetic information of the Sri Lankan politicians, you can see if they are connected to donkeys in the evolutionary pathway, right? So you can find out the relationship between different species, evolutionary relationships, if you know the genetic information. So that can be done, right? So I think you don't need to do that. I mean, it's very obvious depending on what they say and what they talk in the television and all. I think they are related to donkeys. Without a genomic analysis, we can come to that conclusion, right? Am I correct? Right? No doubt, yes. Okay. So right, then the next set of data is the proteomic data. Now we'll see Proteomic data involves the full set of proteins produced by um, a single creature, right? An organism, tissue, or cell, right? So we have, as I told you, so this is the primary structure of the protein. That means the amino acid sequence. We have in proteins, we have primary, secondary, tertiary, and quaternary structures. Primary, secondary, tertiary, and quaternary structures. The primary structure is the polypeptide chain, as it's just synthesized in the protein factory, as we discussed, right? Then it falls into some sort of um, substructures called secondary structures. Main types of secondary structures are alpha helices. We have different names. The helices like this, they are called alpha helix, alpha helices, and then kind of flat structures like this, beta sheets. Now, as I told you before, whether the part of this polypeptide chain is going to make an alpha helix or beta sheet is also depending on the sequence. Now you should be able to analyze the existing set of structures, right? So you can analyze the entire set of 220,000 odd protein structures and the corresponding their sequence, and then see this relationship by using a piece of code. What is the relationship? What sequences are making alpha helices and what sequences are making beta sheets? Can you do that? It's a, you can take 2, 3, 4, maybe 100 structures and try to do this exercise, right? So you can see if there is a general pattern. What we are trying to identify is are there any, or is there any specific amino acid sequence or pattern that makes an alpha helix, and can we identify a specific sequence that makes beta sheet? So how do we do this? So we have to take a good sample of existing structures and then specifically look at the alpha helices. You take, let's say, all 200,000 protein entries in the database and then look at the places where you have alpha helices, right? So for example, now in this particular protein that we looked at before, I'm going to show it again. Right, so we'll put the secondary structure. So there are different ways of visualizing it. Put it into can you see that? Yes. Okay, so now you can see there are different what we call secondary structures. So this is this one over here. I think you can see my cursor. This is an alpha helix, and these flat sheets are beta sheets. If you want to see the actual molecules behind this, I can also put another representation and put the actual molecules. You see, you can see the molecules, why we call it alpha sheets and alpha helices and beta sheets, right? they kind of this polypeptide chain twists into certain shape. It's alpha helix, and here's a beta sheet. Now we were saying that if we could take all the alpha helices in all the proteins in the entire protein data bank, and then we can do an analysis to see if there's a common set of sequences that gives a rise to alpha helices, then how is it going to be useful? Because when we have the brand new sequence for which we don't have a 3D structure, we can predict. Okay, so this particular sequence, according to our previous knowledge, this is going to be an alpha helix, right? So this is going to be a beta sheet. So now here's another example of what you can do as a software engineer in bioinformatics. You can develop tools to identify protein secondary structures. So there are programs, there are algorithms that are written to do this specific task. So there are books even written on protein secondary structure identification, right? So you can introduce AI into this. What is your objective now? You given a sequence, you are supposed to predict the secondary structures in that sequence. From here to here, it's an alpha helix. This part here doesn't have a specific secondary structure. You see here, these portions like still in the chain form. These are called loops. No specific secondary structures, and then these are beta sheets. Okay. Now what I want to emphasize here is where your software engineering skills go into bioinformatics and what you can do and why it is important to do that. That's what we are learning in this course, right? So here's another exercise for you. Take along a sequence and then try to predict the secondary structures. Of course you need some previous information, right? Because when you read it once again, you can have different reading frames, right? Because it's a sequence. You can take, let's say, 3 amino acids, 4 amino acids at a time, 10 amino acids at a time. How are you going to that the frame of 10? You can have it like this, then you can shift the frame, then you have another 10. You can overlap the frame, then you have another 10, right? So it depends on how you going to read it. Okay, so it's a different exercise. You have to come up with efficient algorithms of determining protein secondary structures. Now we go back to our slide again. Now protein secondary structures are there. Then when you have a collection of secondary structures together, we call it the three-dimensional structure. This is the 3D structure that we saw in the protein. This is the functional form. But in some proteins, there are many units, more than one unit like this, aggregating, clustering together, and that is called the quaternary structure. So the three-dimensional tertiary structure is the first three-dimensional functional form, but for some proteins this might not be the functional operating form. It needs to cluster with some other 3D structures like this, and that is called the quaternary structure. Some proteins are like this one over here, we found them in the body, and some proteins we found it like this in the tertiary structure. So now we know what the primary structure is. The primary structure is the amino acid sequence. The secondary structures are two types, alpha helices and beta sheets. Then we have the three-dimensional structure, the tertiary structure. In certain cases, two or more tertiary structures can cluster together to give you the functional protein. So in this case, it has four. So this is given a special name, tetramer. Tetra means four, tetramer. So you have monomer, this is a monomer, single chain. Then you have dimers. So these dimers can be homodimers or heterodimers. Homo means two identical units are together. Hetero means there are two different types of units together. So you have homodimers and heterodimers. You have trimers. It can be homotrimers, that means three identical units, or they can be hetero. They can be three different types of units. So right, you can have then tetramers. So most of the time that's all. We don't see more than 4 units aggregating. So we see about 4 units aggregating. Now we have quite a good idea about the protein structure and why it is important to look at the sequence. It's all related to the sequence over here. That is all related to the genomic sequence on the DNA, right? So this sequence analysis is mainly what we are trying to do in bioinformatics to understand the function of the biological molecules. So the application, drug design, disease mechanisms, and protein-protein interactions. Now, another application I would tell you, the protein-protein interactions. The proteins interact with other proteins in the body, specifically to send signals. We have a signaling pathway inside the body, right? So when you eat too much of sugar, when you take let's say for example caffeine, too much of caffeine, or you take a Red Bull for example, what do you feel? You feel a little bit more what? Huh? What is the word? Energetic, active. So how does that happen? The caffeine in your body, caffeine in your drink goes and binds with certain receptors in your cell. Right? There are receptors like antennas in your cell. Your caffeine goes and binds to these receptors, and then these receptors in the cell, it will start certain signaling pathways, right? Like type of feeling good, right? There's a molecule called there's a rewarding system in your body. The rewarding molecule is called dopamine, right? So there's a rewarding system in your body. When you feel happy, when you stimulate that, you have lots of dopamine, and there are dopamine receptors that will give you this feeling good type of, feeling active, feeling good type of what you call feeling good feeling, right? Feeling good feeling. So what is happening there? So there is a signaling pathway just started by caffeine binding into receptor, a protein, specific protein in your body, right? So it goes down and then give you this feeling happy signals. Okay. Then to do that, these proteins, there are certain types of proteins interacting with one another. This caffeine stimulates one protein, and then there are proteins interacting with one another, giving this message to the correct place, to your brain. Now this is happening through protein-protein interactions. It is important for us to understand how proteins do interact with other proteins. Now once again, the people, if you think, the people interact with other people. We have different mechanisms. We talk to each other verbally. We have what? What is this? Gestures. If you go to Italy, right? What? You will be yeah, no, yeah, hand gestures. So Italy is Italian language. In Italy, that is, I think, the place in the world that they use these hand gestures the most. They have various kind of, as they talk, they have different hand signaling. Then we sometimes, you say that, I don't know, when you say, when you are talking to me in your eyes, and you talk to people, you can use eye communication, eye contacts, right? Different way of contacting. So, but limited way of contacting and communicating. Proteins also talk to other proteins like this. So how do proteins communicate with another protein? Because these are molecules. If this particular protein is there, and then let's say your caffeine binds to this protein over here, then it changes its shapes into something like this, from this particular shape, because the caffeine is bound to it. Now what happens, there is a pocket here, then there's another protein that can come into this pocket, right, and then interact with that. So when a molecule binds to a protein like this, it undergoes a shape change. So that shape change allows another protein to come and recognize, so now there is a signal for me to go and bind to that. So likewise, then when this is bound to that, then there is another protein coming into this one, right? So likewise, the signal is passed down. So you can also look at how and where these proteins do have their contacts. It's once again based on their sequence, right? So protein-protein interaction, understanding protein-protein interaction is one of the main problems in bioinformatics, in biology also. Now there's another application for you to, or an opportunity for you to, as a software engineer, to address this problem. What do you want to do here? What is the problem? You look at all known protein-protein interactions, because there are in protein data bank, once again, there are single proteins. Of course, there are protein-protein complexes that they manage to get the structure for. Then you analyze all of these things and then see how proteins, what regions of these proteins interact with the other proteins, and try to identify and generalize these patterns. Which amino acid sequences tend to interact with other amino acids in other proteins, and what amino acids usually do not come into contact with other proteins. Can you do that? By analyzing the protein-protein complexes. So you are given a training data set, some known protein-protein complexes. You can take them and then, writing a piece of code, analyze what amino acid residues in the first protein are interacting with the amino acid sequences in the second protein. Then you build up a certain knowledge database. Then what are we going to do with this? Then I have brand new proteins. I want to see, is the new protein PX we have isolated very recently, brand new protein. So what are the other possible proteins that this protein can interact with? So you want to search for these specific segments and then find the matching segments on the other protein. You can come to a certain assumption, or you can predict, right, the protein-protein interaction between this given protein and the other existing proteins, right? Here's another application. Now throughout this discussion today specifically, I showed you different places where your software engineering and programming skills can be applied directly to the real life problems in bioinformatics, right? Do you understand that? So it is for you to now think whether you can really do these simple exercises, right? So I think that my job is to show you these places and then give you enough background information so you can do these exercises. That's what we are trying to do this introductory course in bioinformatics. All right. Um, so the other type of data, the biological data we have, is the transcriptomic data. Right? So transcriptomic data refers to the complete set of RNA transcripts produced by the genome under specific circumstances in or in a specific cell. Now that means, what does that mean? It sounds a bit complicated, but it's a simple thing. So you have a healthy cell, and on the other side you have a cancer cell, for example. There's a DNA in the healthy cell, there's DNA in the cancer cell also. Two different conditions. One is healthy, the one is a cancer cell. Now we want to see if people have scientists do isolate RNA molecules that are here in the healthy cells, isolate all the RNA molecules, isolate all the RNA molecules here, different types of signals. So that means different messengers. So if you talk to the messenger, if you analyze the messenger, he is taking the message from the DNA to the protein synthesis factory, as we discussed before. Now the original information, what proteins to be made, is here in the DNA. Okay. Now let's assume in the healthy cell it produces only protein 1 and protein 2. There are thousands and thousands different types of proteins, but we'll take only as an example, it produces only protein 1 and protein 2. But in the, okay, so hold on, let's do it the other way around. So protein 1 and protein 2. In the cancer cell, it produces protein 1, protein 2, of course protein 3. Why? Because the cancer cell is different. So now protein 3 is coded by gene 3 here, gene P3, gene that is responsible for protein 3. Say that in the cancer cell, the gene P3 is overexpressed. It is switched on. Therefore, it gives the message to the RNA and it brings it to the protein synthesis factory, and then of course it produces protein 3. On the other side, on the healthy cell, how about the gene P3? Gene P3 is suppressed. It is switched off. So there's no signal to produce the protein P3. Now instead of analyzing the proteins in the cell, it is easier to analyze the messenger, because if this signal is there, it is definitely inside the messenger, right? So transcriptomic data refers to the complete set of RNA transcripts produced by the DNA. So all the RNAs, if you collect all the RNAs that is produced in this process, if you get that data, that is called the transcriptomic data, right? So why it is important? Because when you have the transcriptomic data, we can see what genes are expressed to what extent. Then not only that, we can also see the expression patterns. When the P3 is expressed, what is happened to the P1? So sometimes this P1 is suppressed. When the P3 is produced, P1 is not produced, right? So this is what happened. This is a relationship. Now you can go back and then relate this to the pathology of this particular cancer. this particular cancer has a different gene expression pattern, right? It makes more of protein 3, and it doesn't make any protein 1. That means your gene 3 is overexpressed and gene 1 is suppressed, right? So we can use that information towards understanding the behavior of this cancer and then finally leading to certain treatments, right? So the transcriptomic data is very important. Now, how do we analyze this is specifically called DNA microarray analysis, right? DNA microarray analysis. So we'll talk about this towards the later stage in the course. So where we take this, here's the picture of it. Right? You take the RNA extractions and then get the complementary the DNAs, and then we convert that into a pattern like this, right? Into a data pattern. Basically what we do is I'll explain it later. So get the RNAs and then add some dyes to color different molecules in different colors, and then we look at the color intensity. Then finally, using a scanner, convert these different colors into numbers, and then we have a formula to rank this numbering and then see the relationship between the genes. So finally, it's a computer algorithm, a mathematical analysis. Finally, that will tell you what genes are expressed and what patterns are observed in gene expression. It's an interesting thing to know, and then it's a detail which we will discuss, maybe next lecture or so. Right. Okay. So transcriptomic data, and then of course the next set of data that we have to deal with is the metabolomic data. So that means the data that is related to metabolism of small molecules in our body. If you take proteins, lipids, sugar, maybe small drug molecule, what is happening to these small molecules inside our body, right? And then people have collected this data and then put it into the databases, right? So that is important to us also. The next one that we have is the did I miss anything? Yeah, metabolic pathways, right? And then system biology type of, I'll give you I think I'm missing that slide. Yeah, I'll try to get the other one in the next lecture, where we look at the system biology of like pathway data, right? Metabolic data and then pathway data. Then the pathway data tells you if you have one particular molecule in your body and all the pathways that are connected to that particular molecule. So for example, if you take caffeine, what are the biochemical pathways that are connected to this caffeine metabolism, right? So then what receptors, and then all these interrelationships between different biochemical pathways. So that's another set of data that we have to deal with. And then finally the epigenetic data. Epigenetic data means epigenetic means outside the genes, outside the genetics. So in the past we used to think that we are our genes, right? So everything that we do, how we look like and all that is determined by our genetics, Is that true? Right? But I mean, of course, there are man-made factors that we incorporate into that. There's a karmic force, there's a god up there who determines what's going to happen to you and all that. That's I'm not worried about that. As far as the biology and the science is concerned, we used to think we are our genes, but now the situation is different. We now say that we are how we respond to our environment. Right? That is the basic idea of epigenetic. That means the genes can also be altered. Of course, I mean, if you are exposed to too much of sunlight, UV light will make your genes change and they will cause cancer. Right? If you are exposed to chemicals, your genetic material will change. Right? So that is how we respond to our environment. There are two interesting books if you want to read. The first one is the biology of belief. The second one is the selfish gene. Two books. The biology of belief is written by Bruce Lipton, who was one of the main scientists involved in the cloning project. Have you heard of Dolly, the first clone animal in the human history? They took a sheep and then clone it exactly, a photocopy of that, and then made another sheep, right? Genetically identical, or cloning. Now the biology of belief is all about epigenetics. Very interesting book. And then the selfish gene. So what is happening to us as far as the genes are concerned, right? So you can see how interesting this genetic information is. Okay. Of course, right now we know about different types of data, biological data, where it is coming from, what are the challenges we have, and then where can we interfere as software engineers in these biological processes to develop certain algorithms and softwares, right? So that is all I have for today. Then we will start with the next section in the next lecture, and then continue our discussion. We will look at the protein data bank file structures, different file formats, right? And then visualizing and analyzing biological data, so on and so forth. Okay, right. Any questions? All good. Everything understood, crystal clear. Right. There could be no questions for two reasons mainly. What are the two reasons? Everything understood, crystal clear, so therefore no problems. What is the second option? I didn't understand anything, therefore I don't know how to ask a question. Which category are you? If you are not on these two categories, you must be having some problems, because you're not going to understand everything that I told you in one go. You need to now you have lots of tools to help you learn, right? You have YouTube lectures, you have open courseware. Do you know about open courseware? Universities like Stanford, MIT, Harvard, right? They have their courses open for everyone in the world for free. All their lectures recorded and available, not only in science and bioinformatics, but also computer science, mathematics, engineering, medicine, all the courses. You can search for open courseware in these famous and leading universities and listen to them, right? Listen to the bioinformatics courses done by other professors at, let's say, at Harvard University, right? And then of course you have online book libraries. Then of course you have your life partner now, ChatGTP, right? Maybe it's not very accurate sometimes, but you can get a rough idea. It's good starting point, right? And of course Google. So I remember when Google was first launched, I was in Canada. They had a banner in one of their conferences. Um, Certain things in life are not to be are meant to be not known. Certain things in life are meant to be not known. For the rest, you have Google, right? So you can. It's very powerful now with even this AI technology and all that. Uh, you have, I think, very good set of tools to help you learn if you really want to learn things. Right? Of course, about Google's, I saw about few years ago a three-wheeler down there in Kovala Junction. At the back of on the back of the three-wheeler, it's written, "I don't need Google. My wife knows everything." Right? So, so maybe people who are married, maybe you don't need Google. You can ask your wife. She will tell you. So now I'm safe because my door is locked. No one can hear it. Otherwise, I also have a problem. Right? So I tell my wife that it's not that wives know everything, but it is all they are also very similar to Google. Why? Because you start typing something halfway through, it predicts the rest of it for you. Right? You are not even completed your search phrase. You only type half of the things that you want, and then it predicts. Your wife is the same. You start talking to them, and then half you are not said the complete thing yet, but they predict the rest before you finish, right? So in that way, I think they are very similar. Google and your wife, for example. Okay, so that's something outside. But any questions? Right. Okay. So I think probably we'll see you guys on Friday evening again. Okay. Then I'll stop here and good night to you.